Welcome to the final part of the final lecture. Up until this point in lecture 16, we've looked at mechanisms of deformation and mechanisms of breakup, and we understand now that emulsions can exhibit both of these types of behaviour depending on the capillary number and the viscosity ratio and the deformation field. What we're going to do now is to look at relative viscosity results and see how we can estimate the relative viscosity of an emulsion so long as we know something about the droplet size or droplet size distribution, the capillary number and the relative ratio of the two viscosities. So we're going to see that when capillary numbers are high, viscous forces dominate and droplet deformation happens. We're going to see that when capillary numbers are low, surface forces dominate and the droplets look like non-deformable entities. So let's start off at that point. Let's look at non-deformable droplets, i.e. low capillary numbers. Moreover, let's look at a result where we have a small volume fraction of droplets. Here on the board is a result that gives the relative viscosity. Remember, that's the viscosity of the emulsion divided by the continuous liquid phase viscosity, that looks very much like Brownian hard spheres. On the right hand side we have an expression 1 plus 5 kappa plus 2 over 2 kappa plus 1 multiplied by my volume fraction phi, and we can see that as we set kappa to infinity, this reduces to the result for Brownian hard spheres. Remember, Brownian hard spheres were for dilute suspensions. Here is an analogous result for a dilute emulsion, and the two are consistent with one another. So this is good, we're happy. So if you have a dilute emulsion, and if you have a capillary number regime, which means that the droplets are spherical, there's a very useful result that is analogous to Brownian hard spheres. Now, as we increase the droplet volume fraction, as we increase phi, but keep the capillary number low, so we're still dealing with spherical droplets, there are two broadly different approaches that one can take. One approach is called a cell model, and I've illustrated that semi-schematically here on the board. A cell model says that, look, you have a droplet, you have a deformation field in the continuous phase that is as a result of the presence of that droplet. The streamlines deform around that droplet, but that streamline deformation only goes out so far. It's kind of a hydrodynamic theory. So in effect, around each droplet there is an influence zone, the zone over which that droplet has influence, but outside of that influence zone that droplet doesn't affect what's going on. So that is sort of the idea behind a cell model. Now, there are a number of different cell models, but one that is quite useful is that due to Jeroen and Gall-Orr, that says that the relative viscosity is 1 plus a function of volume fraction multiplied by volume fraction. Now, that function of volume fraction is actually quite complex, and we're not going into the derivation of this result, but we will look at what this result predicts in a second or two. So one approach is to use a cell model. A second approach is completely different. It's called an effective medium approach, and what effective medium approaches do is approximate the behaviour of a two-phase system as a one-phase system. So we're trying to combine the effects of droplets and continuous phase together as one homologous contribution. So effective medium approaches are again numerous. A key result that is useful is that by Fantine and Pham, and note that the result here is implicit in relative viscosity, so this needs numerical solution. So two different approaches, cell models and effective medium approaches, two different results. Let's have a look now at how these two different approaches compare to one another, and we're going to do this by looking at a plot of relative viscosity, e to r, as a function of viscosity ratio kappa. And we'll look at both of these two models side by side. So here on the board in front of you now are the results from the cell model of Jeroen and Gall-Orr in blue, and the effective medium approach, and they're different. This is for a volume fraction of 0.75, but what one finds is if one plots experimental results on these axes as well, the cell model typically forms an upper bound and the effective medium typically forms a lower bound. Yes, it will depend on the system that you're observing, but there's a generalisation there that sort of stands true. So you effectively have an upper bracket and a lower bracket to behaviour. As we reduce volume fraction, let's say from 0.75 to 0.5, the relative viscosity prediction also reduces. And if we reduce that volume fraction further, from 0.5 to 0.2, again we see that the relative viscosity starts to approach that of the continuous phase. So, two approaches, cell models and effective mediums, and one forms kind of an upper bracket, one forms kind of a lower bracket. Okay. Let's now look at high capillary number. Now, a high capillary number, viscous stress dominates surface stress, and low viscosity ratios mean that the viscosity of the droplet is a lot lower than the viscosity of the surrounding liquid, and so the droplets are easily deformable. So what we have are ellipsoidal droplets. Now, the effect of a whole assembly of ellipsoidal droplets is to increase the maximum packing fraction to a very high level. So phi max can go as high as 0.9 or 0.91 for ellipsoidal droplets aligned in a shear flow. So what we also see are some other physical effects. We see shear thinning. As phi max increases, we get that shear ordering. And remember, we talked about shear ordering in suspensions and said this can give rise to something that looks like shear thinning, because the packing fraction, phi over phi max, decreases with increasing phi max, and that results in a decreasing relative viscosity. Now, elasticity can also be observed in these systems, because what we have are droplets deforming, effectively storing strain energy, because surface tension is present. You're stretching those droplets out, and surface tension will restore them once that deformation has been stopped. And again, we said there is a time scale over which that droplet reformation process can happen, so going from ellipsoidal to spherical. And so that can give rise to elastic effects, even if both liquids are Newtonian, because you've got this shape change and shape restoration happening. Now, there is a characteristic timescale that associates with that elasticity, T subscript E, which goes as the radius of the droplet multiplied by the continuous phase viscosity divided by the surface tension. So we can see for large droplets of high viscosity, we've got fairly long characteristic timescales. So those are the systems where we start to observe more of an elastic response. Now, if we think of high capillary numbers still, so where viscous stress dominates, but high viscosity ratios, those droplets that are non-deformable, look like spherical droplets, that we can assume that the droplets do actually act as hard spheres, and that the maximum packing fraction goes with one of the spherical packing fractions, in this case a random packing fraction of 0.64. Let's summarise a few key points. We've looked at how capillary number and viscosity ratio affects suspension behaviour, and we've looked at measures of relative viscosity. If capillary numbers are low, your dilute emulsion looks a bit like your dilute suspension. Remember, it tends to that Brownian hard spheres result. If your capillary numbers are low and your volume fractions are high, then we've got these two approaches, this cell approach and this effective medium approach, and they act as sort of upper and lower bounds on real observable data. If capillary numbers are high and the viscosity ratio is low, we have highly deformable droplets. These droplets will deform. They will take time to restore their initial spherical shape, and this can give rise to elasticity in a system. If capillary numbers are high and viscosity ratios are high, what you have are behaviours very similar to those of concentrated suspensions.